everyone, it's really great to be here. Can everyone hear me? Or maybe that's better. Okay, yeah, all right. Okay, so once again, hi everyone, it's a great pleasure to be here. And my talk is not going to be about cryonics, but it's going to be about another way of life extension, which is, well, an alternative. Basically, we all are born, and we would like to extend our lifespan as it is right now first. Let's try the biological methods first, and cryonics would be the best action in the worst case scenario. So I'm going to present an experiment which I think is one of the most promising ones that exists right now in terms of life extension. It's gene engineering. So we all know that there already are successes when people introduce mutations in model animals and reach quite significant life extension. Well, for example, you can increase lifespan of a worm tenfold by completely removing the H1 gene. And we do have a human analog for that gene. It's PI3 kinase. And lifespan of mice was almost doubled by suppressing the growth hormone receptor. So, and of course, there are other examples in other model animals. But those animals had those mutations from the very start. So that's genetic engineering. And we don't really want that because we're already born. We cannot be reborn again. So we need to do something about our bodies as they are at the moment currently. And that's what gene engineering, gene therapy does. It translates the successes of genetic engineering into medicine. And this is what we want to do, because we want to cure us, the already born humans. So there already are 4 drugs approved for use in humans, and there are more than 2,000 clinical trials ongoing. And hopefully we will see more and more therapies approved for use in people. So gene therapy presents a really powerful tool to extend longevity and treat age-related pathologies. So this is what we propose, an experiment which will yield a complex therapy that can be used in humans to extend their lifespan and improve health span. So this is just a schematic representation of the experiment. So we will test all mice. We will introduce genes that systemically influence various aging mechanisms, and separately ones that act on the hypothalamus, because this is one of the main orchestrators of human aging. And as a result, we will get a therapy that can be easily translated into clinic. So what genes are we going to test? We know that there are more than 100 longevity-associated genes currently. So we kind of picked the 20 best ones, and we propose to test them. But actually, the actual number of genes, it depends on funding. But those 20 ones are those that create the most significant impacts in terms of life extension in model animals. And we will use adeno-associated viruses to deliver the genes. The choice of this particular type of virus is dictated by the safety concerns. Adeno-associated viruses do not integrate into the genome. They kind of hang in there as extra chromosomal parts. They do deliver the product, those good longevity increasing genes into our cells, but they do not randomly integrate into our genome, by that reducing significantly the risk of getting cancer. Another way how we are planning to increase safety is to the genes will be placed under specific promoters that will be only activated when a particular drug is administered to the system. You can think of it kind of like we can switch on the gene therapy. So you eat the drug and only then you will have the expression. By doing so, we will regulate the duration of the treatment, and this will improve the safety. So we will test old animals because, well, we all are old animals, and that's the fact that we want to achieve. We want old animals to live healthier and longer, and we will use young ones as positive controls, and we'll obviously have the old mice to see what's happening to them. This is the research plan, and the experiment is really easy. So we start with 20 genes, we test each of them individually, and then we find the most efficient and safe genes, and we will test the combinations of them. And the combinations will be of 2, 3, and 4 genes together. We will also test all of the best 5 genes, and the way how we're gonna see which genes are the most efficient ones. We will test various biomarkers, including blood and adipose tissue transcriptomes, metabolomes. So proteome studies will be performed. So we will extensively test aging biomarkers. We'll basically looking at what's happening to those mice, and we will perform safety assessment, of course, because that's a really big concern when you talk about gene therapy. So then that combination will be the treatment. So what are those genes? So those 2 genes that will be delivered to the hippocampus, that's an FKB gene that normally promotes inflammation, and we do not want extra inflammation in our brains as we age, because, well, aging can be seen as a chronic inflammatory process. That's one of the ways to look at it. The uncoupling protein 2 has been shown to increase longevity in mice quite significantly, by 20%. There are certain genes that we want to suppress their activity, but some we need more. So the therapy will be designed in a way so we would suppress the aging-associated genes like the growth hormone or TOR, and we will increase the activity of the good ones like the telomerase, for example, or we will suppress the retrotransposomes. These are the parts of the genome that are pretty much like viruses. They can jump and integrate in random parts of the genome, therefore disrupting its integrity. So we don't want that. We want that activity to be suppressed. But there are other good things that we want to increase, like adenylate cyclase 5 gene. The FOXO genes are really well-known longevity genes. We want more SIR36, sirtuins. You've probably heard about that group of genes. There's evidence that, well, this particular one is really interesting. It looks like we can eliminate senescent cells using gene therapy. So this is the herpes simple virus. This gene selectively kills senescent cells. So why do we think that a combination of of these genes will actually be successful? Well, because previous studies show that if you sort of enter two things at once in a mutant, so if you create a double mutant, if you suppress simultaneously TOR and insulin growth factor at the same time in the same animal, you can have synergistic effect. So this was done by Pankaj Pahi at the Buck Institute, and if you do those interventions separately, suppressing of TOR kinase gives you only 30% of life extension, and suppressing insulin signaling produces 100% of life extension. So if you do all of that together, you have synergistic effect of 400%, and this is great, and this is an indication that some of the genes that we handpicked may create a similar effect. And the other fact that makes us really optimistic is that gene therapy has already been tested in all mice, and it was done by Maria Blasco in Spain. And in that experiment, she introduced telomerase gene into adult mice and really old ones. So 1-year-old mice lived 24% longer, and the 2-year-old mice, which are really, really old because mice live like 2.5 years, those ones lived 13% longer. So it works, and the animals didn't have more cancer. In fact, their health parameters were very much improved. So with that, I would like to say that the aging, the damages that are accumulated over time with aging, there's a quite significant amount of data that suggests that these damages accumulate because of the disruption in the way how our genes act. So this comprehensive therapy is a way to interfere with those age-related changes in the way how our genome acts. So we're pretty sure it's going to work. There's a problem, an organizational problem with this experiment. Obviously, it's not going to be cheap, and aside from the problem of fundraising, there's a problem of basically organizational structure of the whole thing. But organizational issues tend to be more solvable if you have the money. So one of the ways, potential ways how we can raise money for this experiment is using crowdfunding. And even though the task of creating a video for a crowdfunding campaign is quite a daunting one on its own, it's not enough. There has to be a community built around this issue, and this is what we would like to start with. We would like to build a core of like-minded people who would then promote implementing this experiment in real life, well, using crowdfunding or any other kind of fundraising tool. But I think crowdfunding is the most likely one to make significant impact. So we're very much looking for creating collaborations and meeting other people who would be interested in carrying out this experiment. Thank you very much.
Have you thought or attempted to contact the Google Calico effort?
No, we haven't attempted, because in order to contact them, there has to be a clear representation of the project. Basically, that crowdfunding video has to be created beforehand, because you can, to pitch things, you can only have like, I don't know, a couple of minutes, right? So everything has to be in place. There has to be a solid plan. So no, not yet. Yep.
The child, Brooke Greenberg, who is at age 2, stopped aging. I have a photograph of the desk of her at 19, and she's still the size of a 2-year-old, mentally and physically. Have you looked into the genetic aspect of that amazing situation? Do you know what I'm talking about?
Yeah, I know. It has to do with the list of genes that can be picked for this kind of experiment. So that particular case is like a standalone case. It needs thorough investigation, so there has to be a lot of studies performed on this kid. And I don't think that people have really deciphered what's going on there, what's causing the phenotype, like why is she still, why does she still look like a 2-year-old whereas she's much older. So no, that is not in there.
Yeah, she died a couple of years ago, at the age of 22. But I'm specifically interested, would it be a gene that would affect the stop the growing process?
Yes, because we are basically, everything that happens to us is a result of how our genes act. Especially at young ages, especially. That's because before puberty, that's our developmental program that's causing what's happening to us.
What sort of budget are you looking at?
I think well, it depends on the number of genes, of course, on the number of animals, on the place, on the specific lab. But I would roughly estimate it as maybe $2 to $5 million. We are Science for Life Extension Foundation. It's a small non-profit, but yeah, it's just a small non-profit group. But it can be anybody who's interested in that experiment. It's just that we came up with the idea, and we're looking for, people who could implement it in real life together with us. Greg?
Yeah, so this is not your PhD work you're talking
Oh no,
about?
no, that is not my PhD work.
So second question, did I understand correctly that all of your interventions will be targeted to the hypothalamus?
No, just the two.
Okay.
Just NF-kappa B and the uncoupling protein 2. The rest will be done systemically. So the drug would be delivered. Those two gene therapies would be delivered inside the brain. The rest in the bloodstream.
Can I ask one more?
Sure.
Have you considered TFAM, upregulating TFAM?
Yes. So again, this list, it can be enlarged, it can be downsized. It's really quite flexible. So if there's anybody who has a particular interest in gene and who wants to be a part of this, he or she wants to be a part of this project, then sure.
Is your intention, I don't know who you are exactly, but is your intention eventually to form a corporation and have to do other things? What are your goals?
Goal? Life extension.
For this group, I mean, what do you?
Oh, we want the result. We don't care how it's going to be done. If there is going to be, at some point of time, a company formed that would be the optimal way to get the therapy to the clinic, so be it. If it's, but I think for starters, it's going to be an experiment done in the lab, supported by either a crowdfunding campaign or if we're able to find wealthy donors, then wealthy donors. So it's going to start as a non-profit kind of effort. How it will be, I think is for profit, but they're not necessarily. I'm not a specialist on that. Yeah.
What is? Can you talk about how much overlap there is between those genes in mice and people in humans?
All of those genes are in people, of course. Yeah, we all have those genes. They do the same thing in people, so that's why we pick them. They're very conserved, evolutionary conserved genes. Basically, they exist in mice and in us. Okay, yeah. Sure.
Yeah, how is your approach related to what Aubrey Ray does? Is it similar?
It's just an experiment. If some of the genes that would help out with certain parts of sense, like our approaches can be combined. I mean, it's just a method. So.
Or it seems like a promising approach. Are they doing something similar, or you just?
I think they want to do something similar. Yeah, but I mean, I'm going to talk to Aubrey tomorrow, I guess, about that.
New age now, hot summer day.